Hello, my name is Joshua Mugabi. I welcome you to Biology King. In this session, we are going to talk about protein. Draw the basic structure of a generalized amino acid. When you look at our amino acid, it has the NHH, which is the amine group, attached to the carbon atom, which has got a hydrogen atom attached to it and also a variable group. Then it also has a carboxyl group attached to it. So the carbon has got 4 functional groups attached to it: the amine group, the variable group, the carboxyl group, and the hydrogen atom. Outline the structure organization of a polypeptide chain, that is from the monomer to the polymer. Amino acids are joined via condensation reactions to form dipeptides, and a water molecule is produced. The covalent bond that connects amino acids together in the polypeptide chain is called a peptide bond. The polypeptide synthesis occurs at the ribosome via the process of translation. Draw the basic structure of a generalized dipeptide. Now, when you look at the structure of a dipeptide, there are 2 amino acids joined together, one on the carboxylic end and another on the amine end. Water is going to be formed, and they are going to form a bond which is a peptide bond. That is the structure of a dipeptide. Identify the number of different amino acids present in nature and how they are distinct. There are 20 different amino acids present in nature. They differ according to the chemical composition of their variable side chain. That one is the R group. Explain with the aid of a diagram the level of protein structure and indicate their significance. We have 4 different structures that we are seeing here. So we want to look at the structures from one level to another level. We shall start with the protein's primary structure, which you are seeing on your left. That is the protein's primary structure, made up of amino acids, the protein primary structure shows the order or the sequence of amino acids within a polypeptide chain. That is a polypeptide chain with amino acids. This one is formed via peptide bonds between the amine and the carboxyl groups of adjacent amino acids. Primary structure, the one we are seeing on our left, determines all subsequent levels of protein structure. Let's now look at the protein's secondary structure. It is the structure that you are seeing on your left. The folding of the polypeptide chain into repeating arrangements gives us the protein's secondary structure. We have the alpha helix or beta plated sheets. It is formed via hydrogen bonds between amine and a carboxyl groups of non-adjacent amino acids. We look now at the protein's tertiary structure. It is the one that you are seeing on your left. The overall three-dimension shape of polypeptide chain. It is forming chains. It is formed via a variety of bonds or interactions between the variable side chains, or what we call the R groups. These interactions may include hydrogen bonds, ionic bonds, disulfide bridges, or hydrophobic interactions. Let's now look at the protein's quaternary structure, the one that you are seeing on your right. The presence of multiple polypeptides or prosthetic groups to form a biologically active protein. Not all proteins will have a quaternary structure. List 7 functions of proteins, providing an example for each. 1. Structure. For example, collagen. Second, formation of hormones. We have insulin and glycogen. 3. For immunity. There we have the antibodies. 4. For transport. Example we have is hemoglobin. 5. We have for sensitivity. For example, the rhodopsin. 6. We have movement. We have examples, the actin and myosin. Then the seventh, we have the enzymes, where we have catalase and Lubisco. Define denaturation. The loss of protein structure resulting in a loss of biological activity. Identify 2 factors that can cause protein denaturation and explain how this occurs. We shall look at 2 factors: temperature and pH. Temperature. The thermal energy breaks the hydrogen bonds responsible for the tertiary structure. If you look at the pH, amino acids are zeta ions possessing both the positive and the negative region, that is the amine and carboxyl group. Now, for the temperature, the loss of the tertiary structure results in the loss of function. Whereas in the pH, altering the pH will change the charge and solubility of the protein, therefore changing the structure. Outline the relationship between genes and polypeptides. Let's look at the gene first. Genes are sequences of DNA that encode polypeptides. Whereas polypeptides, typically, 1 gene equals to 1 polypeptide. However, exception exists. For example, in transfer RNA genes. Define proteome. The totality of proteins expressed within a cell, tissue, or organism at a given time. Hello friends, thank you very much for attending to Biology King. I encourage you to subscribe.